This video is for The Living Earth, Unit 3.5, Evidence for Evolution. Today's essential question is, what do morphological and developmental homologies tell us about common ancestry? Homology is similarity in morphological features resulting from common ancestry. You can analyze and evaluate morphological homologies in mammalian forelimbs of the human, cat, whale, and bat. As different species faced varying environmental conditions over time, characteristics that were present in an ancestral mammalian organism gradually evolved to enable each species to survive in its environment. Because these species all shared a common ancestor, the bones of the forelimbs exhibit an underlying similarity. Yet each forelimb evolved differently as each organism faced different environmental conditions. For example, the forelimb of a cat has become well suited for chasing and capturing prey, while the forelimbs of a whale have become a paddle-like structure, enabling it to successfully swim in an ocean habitat. But not all structures that look alike or perform the same function are in fact homologous. Similar structures may evolve independently in different groups of unrelated organisms. This results in organisms that appear to possess homologous structures, right, when in fact they do not. A good example of this is found in fish, reptiles, and mammals. Sharks, ichthyosaurs, which is an extinct reptile, and dolphins all have streamlined body shapes and dorsal and lateral fins. All three have sharp teeth for catching prey. However, these organisms did not inherit these traits from a common ancestor. These structures are therefore not homologous. These structures evolved independently as a result of similar selection pressures that produce similar adaptations to a marine predatory lifestyle. This coming together or convergence of similar adaptations is called convergent evolution, and there are numerous examples of convergent evolution. Developmental homologies. Developmental homology is recognized as a common embryonic trait in different organisms. For example, as vertebrates undergo embryonic development, they share certain common traits that can be analyzed and evaluated. These shared traits are referred to as developmental homologies. For example, the early embryos of chicks, humans, and cats all have tail-like structures, as well as structures called gill pouches. The presence of these common embryonic traits suggests that these different species share a long ago common ancestor. As embryonic development proceeds, however, an embryonic trait may be lost and the organism can be structurally different when it matures. Embryos of some snake species, for example, show evidence of developing legs. Yet these legs are lost during the development process. This, along with fossil evidence of snake-like organisms with hind limbs, supports the hypothesis that snakes evolved from limbed ancestors.
The concept of homology refers to structures or genes found in organisms of different species that are believed to have descended from a common ancient ancestor. In general, anatomical structures of different species are said to be homologous if they bear the same relationship to one another. For example, one particular bone in the forelimb of a human, a dog, a bird, or a whale would be considered homologous anatomical structures. The science of developmental homology examines and compares how anatomical structures develop in the embryos of different species. Structures are said to be developmentally homologous if they arise from the same embryonic tissues. It has long been observed that each of the 5 different classes of vertebrates, that is, fish, amphibians, reptiles, birds, and mammals, all pass through similar stages of embryological development. One major similarity is that all vertebrate embryos possess a series of paired pharyngeal arches at one stage in their development. When seen from the outside, the pharyngeal arches appear to be nothing particularly remarkable. However, by looking at this diagram of an embryo's internal structures, we can see that the pharyngeal arches are filled with a complex collection of nerves, muscles, connective tissue, and other structures, all of which will go through rapid developmental changes as the embryo matures. The diagram seen here depicts some of the anatomical structures that each embryonic pharyngeal arch gives rise to in a human being. All of the gold-colored structures are derived from the first arch. All of the green structures are derived from the second arch. All of the blue structures are derived from the third arch. And all of the maroon structures are derived from the fourth arch. This diagram shows that the first pharyngeal arch gives rise to two of the tiny bones of the middle ear. And if we were to examine similar embryological development in fish, we'd find that the first pharyngeal arch gives rise to bony structures that support their first set of gills. So this means that the two mammalian ear bones, the malleus and incus, are developmentally homologous to certain of the bony gill supports of fish, because both of them developed from the embryological first pharyngeal arch.
At this point, you should be able to generate a full and appropriate response to today's essential question. What do morphological and developmental homologies tell us about common ancestry?